Hi, good morning, and welcome to the ZP Developer Zone webinar. So we do this generally on a Thursday at 8 a.m. London time, but we also, when it gets really busy, we do a second one on a Sunday, and this is that second one as well. So a lot of questions have come in. There's quite a lot of material to cover today, so I'll just have to essentially dive straight into it. Don't forget the ZP Academy. If you're looking, even though I'll talk about workshops today, if you're looking for free advice, there is the ZP Academy. There's at least a couple of courses on there that you can sign up for. We do webinars. For example, today I will talk about technical and commercial capabilities as well. We do have collaborations. These are strictly with mostly universities in the UK and Norway because that's where we have our facilities. We do have jobs. I mean, I do have a lot of work requests coming in. We do have the developer zone, and we also have workshops. Especially, I think, on things like CGM, continuous glucose monitoring, for example, which is one of the questions today. The workshops are super relevant. I will go quick and just say that we've got these workshops. They're happening in November in Norway and Coventry. If a link jumps up, I'll put the link underneath the video under YouTube. And we do have one in December in Norway. And that one in December in Norway is a 2-day workshop. It's very comprehensive, and we will cover aspects like CGM. There's also free webinars. So today I'll talk about nitrate sensing, ammonium sensing, TAS sensing. All of our technologies, or a lot of our technologies these days, again, if that link jumps up, then that means there's already underneath. I have been asking people to sign up for this. So people do come to the Julie webinar, because actually that will cover in the end what we describe ourselves at ZP as sensor to API, so sensor to application and program interface. So even if you're working with us, we will be, for example, putting the data in the cloud. If you're using our technologies, the data goes to the cloud. So there's a Julie webinar that would be good to come to. Critically, for a couple of questions today, there's the Sense It All webinar. So the guys who are trying to do detection on the breath, or the guys who are trying to do agriculture, please come to the Sense It All webinar. And then if you want to see an application of Sense It All, please come to the Food Sense webinar. These webinars are free. The link is here. And we do one in the morning and one in the evening European time. And that means then that we can cover most parts of the globe. So I hope people will sign up for that. It's probably worth saying that if you're interested in the commercialization of electrochemical assays, which if you're in academia or a startup, we are doing a conference on that in June in the UK. There is an early bird registration, and this is the website as well. Again, I apologize for speaking fast today. It's just that there are quite a lot of material to get through. We do on-site workshops for people as well. We call these kind of corporate and consulting workshops. So on a workshop, for example, we can come on site and we can do things like construct and test the generation 1 glucose wire sensor. If that's not apparent what that is, then as I go forward, that will hopefully answer that. Construct and test the Gen 2 glucose sensor, test the ketone sensor. I'll just read these very quickly. Learn the theory of Gen 1 and Gen 2 glucose sensors, ketone sensors. Discuss all the analytical techniques that are out there, like amperometry, voltammetry, potentiometry, impedance spectroscopy, and how they're used, for example, in CGM, continuous glucose monitoring. By the way, depending on the attendees, if it's more like agriculture or IVD, in vitro diagnostic, we do change the scope as well. We are able to do with that. Talking about commercial applications, critical parameters, how to achieve factory calibration, which is important to many sensors. And in fact, that will be some of my slides today. Is you'll notice that ZP, when we're doing work, we never do, well, at least in our own products, we're not doing in-field calibration. How to produce sensors at high volume, discuss ion selective electrodes, discuss QMS in high volume production scenarios, and discuss project management, because obviously at ZP, I really think it's just program management, because we don't do projects, we do programs over several years in order to get people to market. Our philosophy at ZP is what I would call lean. So there's really an end user. So that end user might be a farmer, or it might be a patient. So we kind of have this philosophy that there is somebody with a real world problem. And what's nice about ZP is we've set it up so that, in fact, many of the elements in order to help these people, whether it's nitrate for the farmer or nitrate on the breath for the patient, we've actually set up many of these elements, so you can consider yourself, a scientist, an engineer, maybe an entrepreneur, maybe a business person, and so we've tried to put elements in place, things like the Sensi All platform, that will allow you to get products into the hands of these people as quickly as possible. At ZP, we do follow a kind of lean startup mentality, which is, essentially trying to get things done as quickly as possible. I understand you can't have a minimally viable product in the context of healthcare, but you can start, trying to give patients and nurses and clinicians technologies to see, their response, let's say. So we're pretty good at getting from you on the left-hand side to people on the right-hand side who could really benefit from this. The technology stack that we have means that even if you give a patient or a farmer a sensing technology, the data will immediately go to the cloud, and that means you can immediately look at it, and it means you can actually start, confirming that it's working the way you would want, and also kind of any kind of improvements that need to take place. So you're kind of in this cycle, and that is, I would describe as a kind of lean. The term used to be lean startup mentality. But I would also call it lean research these days as well, that we need to bring some of that lean thinking also into research and development, and maybe even into academic research and development as well. So at ZP, our whole philosophy really is just to get people from ideas or where they are at the moment to actually testing with real world people, and then getting the feedback as quickly as possible. Our entire technology stack does that, and that will be part of my slides today as well. So just to get to questions, so we do these webinars once a week, or sometimes twice a week, because we're trying to help, we're trying to answer questions that come in through the week. So somebody's interested in expanding their platform beyond SARS-CoV-2 in order to have other ZP sensors on their platform, which I'll talk about. Number 2 is really about sensors for agriculture. They definitely had nitrate listed in their sensors for agriculture. Question number 3 is really about CGM. Have you studied the compatibility of your sensors with whole blood? The answer is we have, but we've done it with paid clients, and some of that data will be owned with those clients, let's say, and we won't necessarily discuss it. Questions like, what's the expected sensor lifetime? If it was a glucose sensor, we've had glucose sensors running out of lab for 9 months continuously. Do your sensors need to be calibrated, and how does that work technically? Well, there's really 2 mindsets. You can do factory calibration or single point calibration in the field, and that's to be determined. Even though ZP might have a CGM sensor, and I suppose it's worth saying that, I started off my glucose diabetic work in approximately 2000 when I was with Abbott, making glucose strips in those days. I was in California for quite a few years, and I worked on CGMs in California. In those days, CGMs used to do a finger stick test when they first started to be used. So they were kind of doing single point calibrations. In fact, they would do glucose testing on the patient for the first few days whilst the sensor essentially settled down. These days, they've gone calibration free because they're actually much stronger on their manufacturing. So calibrations to be determined. It really depends on the final application, which I will touch upon. Accuracy then is really linked to manufacturing and the application and the calibration routine. So there's 3 things that really come into play there, even though we're held at 4 fingers. There's no sensor delay. These glucose sensors are very fast to respond. I will discuss that. I've got some data later on this in this slide deck. How much do sensors cost at scale? I'll give you a link to a website where we show that if you're starting to get things to like 1 million, let's say, then depending on the manufacturing technique that you use, then you can get closer to €1 per sensor. We do have a lot of microneedles as well. It's probably worth saying that on microneedles we have a patent family or patent portfolio of 50 patents, so we are particularly able to lend into microneedles a freedom to operate because of that patent family. Question number 4, somebody's very interested in our single purpose biosensor circuit. I'll just say it now and I'll say it at the end as well. They are asking whether they can multiplex from the single purpose biosensor circuit. I absolutely had a conversation with our guys, a couple of electronic engineers, and they reminded me that we have tried it. And the reason that we sort of, at the time, we didn't have time to finish it because, in fact, we were getting some crosstalk between sensors when we try to multiplex. So I'm not going to be able to give you a Gerber file or a design for multiplexing on that single purpose biosensor circuit. That is something you're going to have to work out. I did speak to them about integration with LabVIEW, and they said, yeah, I mean, it's straightforward. You should be able to do that. But I think the first instance for you is to get one of these. I did give you the data sheets. If you go forward with one, then come back and ask us some questions. Then I would suggest, just for interest, we do have a. This is the set of electronics. We use this throughout our products. So if people are interested in CGM and are wondering what the kind of electronic size could be, the chip that's held between the two fingers there, that is a fully functioning, essentially potentiostat. We use that in even our Sensei All platform. So we're very good because we've got this consistent set of electronics. So we're really skilled with it, and we know that it can, work obviously for CGM, for ketones, for nitrate sensing, for total antioxidative status. It's the same electronics throughout. Question number one, they were particularly interested in measuring analytes on the breath. I think this is just more for interest. There is a company out there called Exhalation Health. It's no secret that we've been their development partner over years. This is CE marked. It's got the capability for doing COPD, chronic obstructive pulmonary disease, for which they have a large patent portfolio. So detection of biomarkers for COPD. These guys have a good patent portfolio on it. And they also did a nice piece of work on SARS-CoV-2 during the pandemic and even got it onto the market. Of course, SARS-CoV-2 now is, it's not a sort of a super important analyte at this moment, though it has been globally significant. Things have moved on a little bit as well. But I suppose this is a nice example at ZP. It's not all science and R&D. Actually, what we mostly do is actually commercialization of products like this. I mean, this is, as you can see, it is a commercialized product and is pretty good. So they want to detect several analytes, either ambient air or exhaled breath. What's nice about this, I had a look at their papers, and all of these produce basically what their value add really is the condensation or the bringing of vapor into liquid, let's say. So having read the papers, and I've only showed these for educational purposes and for discussion, they can either do it from the breath or they can do it from ambient air, but either way they produce a drop. Now the thing that struck me is here that they have a screen printed electrode here, and I don't believe that they are using our screen printed electrodes. I think I recognize the manufacturer on that. So I would appreciate if they did change onto our screen printed electrodes, because we do have a whole range of screen printed electrodes and they're really good. It comes back to the CGM questions earlier on about manufacturing. That what's really good at ZP is once we mature manufacturing, there's a difference between manufacturing and matured manufacturing. Once we've matured it, which we would have on our screen printed electrodes, and I'll put the links here, they're really reproducible. So you'll see us, and I did say please come to the Food Sense webinars. You'll see us do these webinars. We do live demonstrations of how our technology platform can be used in the real world. This is the one for Chile. I'm just checking the thing there. And what you'll notice is no calibration, and that's because of the reproducibility of our manufacturing. I must say it's easier to do non-calibration in a IVD application, in vitro diagnostic application, which this is really, rather than if you're trying to put a sensor onto a human body or into much more complex samples like tear fluid or blood or urine. Those are more challenging applications. Application like this is actually fairly straightforward, so we could do no calibration there. So I don't want to make that a blanket statement, but what I do want to say is the underlying screen printed electrodes that we use in many of our products, including when I look at the technology from that university, use our screen printed electrodes, because then you're going to have a much more reproducible electrode to start with. Because when we make our things like screen printed electrodes, we make sure that actually they're all electrochemically tested. If a sheet like this doesn't have enough reproducibility on it, we will reject it. I mean, this is sheet 1, and you often don't ship sheet 1 because sheet 1 is kind of essentially your setup sheet. But we will take sheets from the production run, test them by an electrochemical method, look at their mean signal, look at the standard deviation, look at the relative standard deviation. Rule of thumb is you want your relative standard deviation in manufacturing to be less than 10%, so that it can be useful either clinically or useful in the field. So we'll check, make sure that our relative standard deviations are single digit. This is done for several sheets in a production run, and we'll sort of track the production run as well and make sure that everything is good. It's that underlying reproducibility that then makes it a much better sensor when you build upon it. So even though these people are asking us about our biosensors, I noticed that you are using screen printed electrodes. So I don't think you're using ours, and I appreciate if you did, because I think our reproducibility will make your life a lot easier. This is some actual data. We just go into the lab often with this kind of work and just actually test at different concentrations, and then we do get signal versus concentration quite nicely. And you can see our, in this case, there's few sources. There'll be the application of the sample, and then there'll be underlying sensors. So they do produce a variation, so it's real data, but you can see that we can easily separate on concentration there. So quite happy with that. Back to the question in hand. So these people are, what I'm going to suggest is they want to test some analytes. They had nitrate and TAS. So when you've got a whole list of analytes, please start simple. Just start with one. I know you've already done the SARS-CoV-2, but rather than jumping into a whole new panel, start with one or two just to kind of make sure that you're comfortable with our technology. Because what you have here is your value add for me is the condensing of that sample, and then you produce like a droplet that there kind of goes onto the sensor. So we've got something called the Sensi All platform. There is a webinar for that on the 13th of December. I hope you sign up for it. I will have put links underneath the video. You can take that sample and put it on our Sensi All platform. This QR code, if you scan it, I'll pull myself out of the way. If you scan it, it will show you that we have an app for the Android on the Play Store, on the Google Play Store. And you're going to find that's a very powerful app, because then that app will link up with this hardware. And this hardware is the Sensi All platform. So the Sensi All, it has LEDs, which kind of describe the status of the machine, whether the machine is on, when it's got Bluetooth connection, when it's doing the assay itself, and when it's completed the assay. So it's got these kind of traffic lights on the front of the instrument. So it really gives a really good user experience, which comes back to my lean manufacture, not lean manufacturing, but my lean startup or lean research comments earlier on. This is much more productized. Now, I understand it doesn't integrate straight away with your condensation platform, but at least half the job is now done, the electronics, and I appreciate you've got electronics as well, but I think your value add is not on the electronics. It's actually the sensor and the way of bringing sample to the sensor. Those are the value adds. When I was in California, people would, we would say, look, the electronics can always happen. Just assume that. It's actually the preparation of sample and the sensors. These are the real value adds. This is where the IP will be, not on the electronics themselves. That's why we've made these kind of electronics generically available, because that's not the value add in research. We have this Sensi All platform. So I think you can condense your samples, put a drop on this platform, and off you go. It's probably worth knowing about this hardware, like I say, coming to the webinar as well, but the hardware is pretty elegant, has a really nice sort of user experience with the LEDs. Just little features like, if it gets contaminated, and this was also relevant to question number 2, is asking about agriculture as well. If it for some reason gets dirty, they can actually just dismantle it like that, give it a wipe down, and reconstruct the device. Come to the Sensi All or the Food Sense webinar, because then you'll see the whole workflow in place. And there's a really important part of the workflow, which is we put all the raw data then that comes off this into the cloud. So you can actually, obviously, A, you can get at the raw data, and B, if you've given it to a farmer, or you've given it to a patient, or you've given it to a clinician, when they're running this work remote from you, it will actually put the raw data into the cloud, and you can see it, and that's really important for things like quality control. So at ZP, we have built a Sensi All technology stack, which genuinely moves all the data, all our sensor data, via the electronics, via the app, which the QR code will jump up again in a minute, all the way to the cloud. And that's really helpful for you, especially part of this kind of lean research, especially like I'm thinking about nitrate sensing. So you're trying to do nitrate sensing in an agricultural setting. Agriculture, really should be remote from you. It happens on farms. And so people can go around testing for nutrients in that case, and actually the data can end up back in your hands through the cloud. And also the Sense It All platform is very flexible. You'll see in this video here I'm holding against different QR codes. Every time I go to a QR code, it actually changes the target analyte in that case. But I'll expand upon that in a minute. So ZP, we've got a whole host of electrochemical sensors. I'll put the link here, but most of these are then compatible with our Sense It All platform. I'd almost say all of these are compatible with our Sense It All platform, including ORP, oxidation reduction potential. Also glucose is up here, pH is up here, etc. Sense It All, this is that QR code again. So if you're curious about at least seeing that we have this app available, scan that code, and then you'll see that in fact these people are particularly interested in a whole list of analytes. I'd say reduce it down a little bit. Do go after nitrate, go after total antioxidative status. The reason being is that those QR codes, you're not interested in chili, but that code exists. You're not interested in ginger, but that code exists. By the way, I haven't asked you to do it, but if you scan these codes, you would find out that actually you were able to change the app would read the QR code and then change its target. But we do have nitrate on there and we do have TAS on there. So those two are kind of good to go, let's say. It's probably worth saying that, slight side comment, but we have spent millions on the nitrate and the TAS sensors, by the way. These are very matured technologies. We do have our Sense It All platform. I regret that I don't have a link there, but I think a link will appear later on anyway. So nitrate is something that we have on the website: chloride, phosphate, potassium, pH, nitric oxide, uric acid, hydrogen peroxide, lactate. But my genuine suggestion is go for nitrate and go for TAS as a starting point. When people go too wide too soon, they give themselves too many problems or too many unknowns. I would just say condense your vision, let's say. So we have the TAS sensor and the TAS meter. I'll put a link there, and we also have the nitrate starter kit as well. The way Sense It All works, I'll run that video whilst that's happening. But we have, for example, the TAS sensor. I'll use the example of TAS in this case. As I say, it's had a lot of money spent on it, so it's a very matured product. It works with our Sense It All platform. You can condense. It also works with the app. You can download the app now, but the app obviously is really waiting for a Bluetooth connection to the device, which you don't have. But it is compatible with Julie. So you put the sensors into the hardware, you put the sample onto the sensor. If you scan the right QR code, then the meter will know what application to run. For example, TAS. It will run the meter. The meter will give the data via Bluetooth back to the hardware, and the hardware will upload to the cloud, and then it will give you a relevant concentration depending on what the analyte is. Now, what's nice about that as well is you get all the raw data in Julie, so you can go in there and visualize it. So if you really are trying to treat patients and you want to do remote work with these patients, which I think you should, you don't put the name of the patient or the patient's age in there. I understand all about HIPAA. You just put a test ID. So I do understand about, you do not put patient data just into a cloud willy-nilly. But you actually get the raw signal, and that really helps you with quality control and make sure everything's working okay. What's nice about that, especially for startups as well, is that people like ZP then can, if you grant us access, we can also see the data as well. And so I'm supporting clients at the moment in India where they're doing the testing. Data just goes straight to the cloud. They ask me, is it working all right? And I can just, as long as they share what's called a cluster with me, I can see the data. It's so smooth. That really works really well. Question number 2 is really about agriculture. So I'm going to come slightly off doing analysis on exhaled breath and now come on to agriculture. They're interested in nitrate, ammonium, pH, oxygen, ORP, oxidation reduction potential. I'm surprised you haven't got phosphate and potassium in there as well, because the NPK panel is pretty famous. But we have been working on this, really starting, it might have been 2019, maybe beginning of 2020, because it was definitely during the pandemic. That we started doing this. So we've been doing this, I think in January 2024, we've been working on this kind of stuff for about 4 years and really put some millions into this. At the moment, they've got the pH sensor. No worry about the shelf life. Don't worry about the shelf life. Those pH sensors are really quite robust. So I wouldn't be super concerned about, you've got the pH sensor good. Is it going off? No, I doubt it. Unless you're leaving it in direct sunlight at high temperature, I suspect you're not, and therefore it should be okay. You're interested in nitrate and ammonium and pH, which you're doing already, uric acid. Oh, I've actually added uric acid because people in farming, I realized I put uric acid. I meant to put urea. My apologies. We do have a urea sensor, so my apologies. I think one of the biggest fertilizers used in the US is not so much nitrate. I think they're using ammonia and urea, so my apologies. But we do have a urea sensor as well. And you're interested in ORP. ORP, oxidation reduction potential. I can understand that might be useful for understanding the kind of health of soil. There's also something called TAS, total antioxidative status. Google TAS, total antioxidative status, because this is ORP and TAS. I think they really end up telling you the same story, and I would actually go for TAS because we've actually put a lot more effort into our TAS sensor. So then there is a nitrate starters kit, and I realize I haven't put a link here, and hopefully I'll put a link underneath. So don't start with all of those panels, just start with something like the nitrate, and then that nitrate kit is cloud enabled. So if you do some testing on that nitrate sensor on that kit in your lab, the data will immediately go to the cloud. You can see your own data, and if you've got questions, you can also share it with us. And once you've kind of got it, the application working, you can then actually send it out to farmers, and you can ensure that they're having a good experience as well. Question number 3 is around CGM. I know I did flash through this earlier on. I'll just do a quick reminder. We have done whole blood testing at ZP and with all our sensors. We generally do it with clients. These are not, it's worth me saying that sensors that are available on the ZP website, we generally make those available for R&D purposes only. I do have some special agreements where some people are buying it for medical applications. But otherwise, they are for R&D purposes only. What is the expected sensor life? We've had sensors running in the lab for over 9 months. I know I've said that already. Calibration, how does that technically work? It's really a system question. You're asking me a sensor question, but the system question is when you put the sensor into the application and there's electronics and it's in that real world, you really have to do quite a detailed discussion about the risks and the pros and cons of where you do calibration. And I actually have a bit of a roadmap on this kind of stuff that I kind of say that the amount of calibration that you need to do decreases with time and as the project matures. What accuracy can you require? Well, I mean, in the end, I suppose it doesn't matter what accuracy I say. You need, in order to have CGM applications, I think the MARD, the mean average relative, difference these days that they're expecting is approximately 8%. What sensor delay? Well, there's raw signal, the sensor responds in seconds. How much to cost? Euros. And I'll talk about the microneedles as well. So ZP, I've been making glucose sensors for 23 years. Also did in California, doing CGMs, and then we launched ZP in 2014. We are ISO 13485, so we are certified for the development and manufacture of electrochemical biosensors, including CGM, and that's what we do. We have a large-ish facility, and we're in Norway and UK. This is our facility. It's not labs. It's not the kind of standard labs. No, these are clean room facilities where we do clean room manufacturing. These are the buildings. We do wire sensors, microneedles, planar electrodes, sensor characterization and testing. This is specifically with CGM. Electronics for CGM, jigs and rigs. People really forget, that you need a lot of test kit when doing this kind of work. We also do multi-analytes, ketones, glucose, for example, lactate. We do documentation when we're on a project in line with ISO 13485. We also do aseptic manufacturing. It's probably worth saying that when you put sensors on people or near people, then you've got to kind of do a risk assessment that they're not going to get infected by those sensors. So that's work that we've done, and we have a clean room for it. And we do these kind of wire sensors, and we also, so for example, this is the complete sensing package with a wire sensor. This is the wire sensor here. This is the electronics here. That package length here is about 38 millimeters, not super optimized, because we didn't need to shrink it down in that particular case. But if you were trying to make a sort of transdermal we even put that sensor in a flowing bloodstream. That would be kind of suitable for that. I have seen people do that where they've taken these kind of wire sensors and put them in. When I say have, I was involved in human blood. And just a warning, but what we do with these sensors is we do operate these sensors actually into fish. So I think that's quite a tough application, and these sensors kind of sit in these fish. In fact, I don't have the video here, but the fish, this is anesthetized. The fish swim around with these sensors in them. I'm just going to change course slightly and talk about CGM. ZP does have quite a lot of patents around CGM, and we have a patent portfolio, a patent family of about 50 patents. It's probably worth saying, I mean, you know, these sensors, I mean, the way they're packaged and stuff like that is because they were actually going into human trials. And we've just got a lot of experience on making microneedles and functionalizing microneedles. Got a lot of experience on testing and also testing for interference. This is linked to question 4, but everything we do, we in the end, it all comes down to one set of electronics. And this electronics is really mature. I mean, ZP started making electronics. I was obviously working on electronics for biosensors pre-2014, but from day one, 2014, we were starting to make electronics, and now they've really matured up, you know, including the shielding and stuff like that. And, you know, we use them on CGM projects, ketone projects, nitrate projects, ORP projects, all of it do that. Jigs and rigs, it's not enough just to kind of make sensors, you know, so much effort actually has to go into how do I make electrical connection within a reproducible fashion, even in my laboratory. So many CGM projects just suffer from poor quality of data, and because they don't understand the rigs and jigs requirement. This is quite a funny one actually. I didn't realize this was going to play, but this is us just testing microneedles in an interstitial fluid system by pushing it through as you can tell, it's not a real arm, but we were testing it there. So I mean, this kind of work really takes some investment. This is the ketone sensor. Here I can see that the ketone sensor is slower to respond, and their question is how fast is the glucose sensor. The nice thing about ZP is I know why it's slow to respond. You know, it's not like it's a mystery to us. This is actually a ketone sensor and a glucose sensor running at the same time. So the glucose sensor, when it's responding to glucose, it responds really quickly. It's up, you know, in a few seconds. If you're wondering why I'm not going to get into this now, but people talk about transdermal ISF sensors. There is a lag between blood and ISF, but this is not that place to start discussing that. But we can put glucose and ketones, for example, together. We could also put lactate together as well. I need to go a little bit faster. Yeah, let me jump forward a bit more. We are ISO 13485, so we are able to, and people try to skip all this documentation and just go for something, but in the end it'll bite you. So unfortunately, a lot of your budget when you're doing this kind of work has to go into proper workflows and proper documentation. Otherwise, you don't have it at the end. We do glucose fabrication in a 100-meter square clean room with class 5 zones. In fact, she is testing some glucose sensors in that particular image. So I know I've said it already. We are ISO 13485. We make glucose sensors CGMs in planar format, wire format, and 3D format. It's probably worth saying that even though ZP can get our technology readiness level quite high, and we can also get our manufacturing readiness levels high, the reason I'm sort of reluctant is every time somebody comes to us, they're really coming in with a new application readiness, a new application. So in this case, these people have a particular vision, and then the questions about how the whole system will perform is really a question about, you know, ZP can make good sensors and we have good technology. How it performs in your system or your application, you know, that I would really think that you're at the sort of ground zero in that respect. So I don't want to overpromise on us because a lot of it depends on you. It's kind of interesting actually because in glucose sensing there's what they call generation 1 sensors, generation 2 sensors. There is also generation 3. I really wouldn't be too concerned about generation 3. We've worked on them. I don't think they add a lot of value. But the reason I bring it up is because some sensors on the market today are generation 2, some sensors are generation 1. The accuracy that these companies achieve, Dexcom claim that they are have the best MARD on the market, as I say, of 8.2%. I mentioned 8% earlier on, the mean average relative deviation. So they say that they're the best on the market at the moment, but they're actually a generation 1 sensor, where one of their competitors is a generation 2 sensor. So it really is, even though that some people would argue generation 2 is better, the accuracy achieved by Dexcom is not because of the sensor, it's because of the sensor in combination with electronics and in combination with the algorithms that they run, etc. So it's not a simple answer to say what is your accuracy. We do manufacture CGMs in several formats. I think the format for these people should be going for is planar. It's easiest to scale, and we can comfortably make 100,000 per month. You can do wire assembly. I have seen people use this kind of assembly as well in the application. And then there's also 3D microneedles as well, which we have the strongest patent portfolio on. The commercialization of a CGM with ZP starts with, you can start with consulting by the hour, micro projects, mini projects, or proof of principles, or major programs where we're really talking about 500 hours from ZP per month for a couple of years. And I've put some links here so you can kind of find out. If you want to start on your own, you can buy CGM sensors in that link there. And then the Sense It All kit is also in that link there as well. Question number 4, last question. My apologies for this. So we've been having a bit of a long conversation on this. So for some of you, this might seem, what's he talking about? It's very quickly, somebody was very interested in our electronics. I've suggested their electronics. They asked me for a data sheet. We've given them a data sheet. They are interested in me answering whether they can use it with LabVIEW. The answer is yes, but I will not be able to give you direction on multiplexing it. So there's one chip that they're clipping onto the board here, and you can buy that board and etc. That one chip will do one sensor. If you want to multiplex, the guy said, yeah, anyone with electronics experience should be able to sort that out, but we've had a go ourselves. We didn't put a lot of time into it, and we didn't like the crosstalk that we were getting through the multiplexer that we were making, so therefore we can't suggest a multiplexer because we haven't finished solving it ourselves. That was the old version. You can see we've come a long way since then. I mean, that was completely exposed. And you can buy that here. So this week we've answered 4 questions, and a lot of it comes down to the Sense It All platform. Well, a lot of it also comes down to people are asking for ranges of sensors. I say focus in on the thing that's most important to you and work on that. I suppose it's worth saying that, at CGM, on the CGM front, ZP has put in, I don't want to be too hyperbole on this, but easily we've put in something like 10,000 hours for the last 5 years. So it's, over 50,000 hours on CGM. We're really set up for it. Last question, number 4, we're not multiplexing at the moment. I aren't able to offer you a Go profile. So I want to say thank you. If you've got any questions for ZP, don't hesitate to reach out to us. You can see this is a long slide set. It might have been something like 84 slides. So you can see that we do have to put a lot of effort into answering all these questions. And it's just finding the most efficient way of doing it. Each one of those questions is kind of almost like a 2-hour meeting in itself. So I try to put together as much as I can to try and do a bit of a brain dump to get the conversations going. Okay, so if you've got any questions on ZP, don't hesitate to reach out to us. If you want to have a private conversation, you can also consult with us. Otherwise, take care. Okay, thanks very much.